So a lot of you have had, to one extent or another, this particular subject area before in terms of mitosis, and eventually we talk about meiosis too. And meiosis 1. Mitosis is technically nuclear replication, production of new nuclei. Since the vast majority of the time, the reason a cell does that is because the cell is going to make another cell and move each of the two copies into each of the two new cells. It becomes kind of synonymous with cell division. So, but every once in a while you'll run into just an issue where that definition becomes important. Most of the time it's not. Now this pie chart, none of the pie charts that you can see are accurate for every cell that does this. Is that this one has this huge cycle where the division part is a very small part. And for cells that are, say, in an early embryo, is that these would be much more similar. Here they're just showing the wedge for mitosis is bigger. But don't get locked into whatever image you have in your head saying that these are the times. Also, if you've learned this before, typically they give you telophase as when cytokinesis occurs, cell division. That is a lie. Is that it can occur in telophase, and a lot of times it occurs in telophase, but basically once you get the chromosome copies away from one another, you can divide the cell. And in plants, they tend to do that pretty doggone quickly after they get the chromosomes away from one another, so they divide in anaphase. And for a lot of cells, they want to basically get reconstituted before they go to the trouble of dividing the cell down the middle. So they're actually back into this interphase stage before they actually divide the cell. So when the cell has separated its chromosomes, you're in anaphase. We'll cover these phases. And then you've got kind of a reconstitution phase of telophase, and then interphase is the cell doing its job in between divisions, nuclear division stage between mitoses. And at any of those stages, you can divide the cell. You don't need to do it. I mean, it is, for a certain type of cell, cytokinesis is going to occur in one of those three stages. And it'll, if it does it in telophase, it'll always do it in telophase. If it does it in interphase, it'll always do it in interphase. Okay, now that I've confused the hell out of everything. We're in interphase. There's a very small population of cells in our bodies that are not in interphase right now. And that's because of the most important function of interphase, which is the function of the cell. Cells have jobs to do. They don't do it while they're dividing. They don't do it while they're making a new nucleus. They do it in between. That's interphase, inter for between. That's why they call them interstates that run between states. So, and some of our cells have been in interphase since we were late stage embryos and are going to stay in interphase until they go into dead phase when we die. Your nervous system cells virtually never divide. Your skeletal muscle cells virtually never divide. I think your smooth muscle cells don't divide either. Your cardiac muscle cells divide for a while when you're a kid as the heart grows, but pretty quickly they stop dividing and just start to get bigger. That's true for most muscle cells. And there are certain populations of connective tissue where once the cells are in place, they can grow, but they don't make more cells. And of course, if they're not making cells, then they are doing their jobs and they're in interphase. So the two major functions of interphase is the cell does its job. The other major function only happens in cells that are actually going to make new nuclei, probably new cells, and that's to get ready for it. So we've got this pink interphase divided into a G1, an S, and a G2 interphase. For our muscles and our nervous cells, that sort of thing. I'm having one of those days. The G1, the G is to signify growth. Because once you've made a cell, it's only a fraction of the original size. It needs to get back to the right size. So it adds bulk, it grows. Adds function too, typically. And here to here is what your muscle cells are doing. Is it because they're not planning on dividing again. These other two sub-stages happen in cells that are heading toward that. And that's the other function. They prepare to divide. Why is it called an S stage? Because it's making stuff. It's making copies of the DNA. It's making copies of the microtubule organizing centers that are going to produce the spindle. It's making basically everything it's going to need to get through the divisions, because going into the division, all of the DNA is going to get packaged very, very tightly, and it's not going to be accessible anymore. You can't make stuff when you have a visible chromosome. It's packed up too tightly, you can't get at the codes. But after everything has been made, you do need some time to get everything into place. So the G2 stage is really the, okay, we're ready to go, we've got everything, let's put it on the trucks and get it out to where it's supposed to be. So in cells that are going to divide, you have these distinctions. In cells that are not going to divide, they're just kind of stalled. Oops. Sorry, I'm swapping around some of these things. So these things are kind of in idealized cells, or cells that are normally used in a mitosis lab, which I think we're doing this semester. Because it's a weird semester, I don't have a clear picture in my head of what all the different labs are. We're slipping in here, but I'm pretty sure we're doing a mitosis lab, where we look at plant cells dividing and animal cells dividing. So you have interphase. Because this is when the cell is active and using its DNA, the DNA is spun out. Now, even spun out, it will typically pick up DNA attractive stains, which is why usually when you see cells under the microscope, the nucleus is a different color than the rest of the cell. Could be dark blue, that's very typical. It could be red. There's a whole bunch of different stains that will stick to DNA and make the nucleus show up. But because the DNA is kind of diffused throughout the whole nucleus, you just get kind of a color. Now, what also may show up in the nucleus is the nucleolus or nucleolus. And there may be one, there may be multiple ones. They may be too small to show up in a light microscope, doesn't resolve them. It varies from cell to cell. Now, here's your microtubule organizing centers. Your centrosomes / centrioles / kinetochores. They don't all mean exactly the same thing. They mean bits and pieces of each other. We're going to call them centrioles. Centrosomes is kind of more the overall molecular complex, and kinetochores is more the technical term for it. Weirdly enough, inside you've got one circle of microtubules oriented this way and another one oriented this way. That's very common in microtubule organizing centers. But these guys, You've got one from the original cell that you divided from. You got to make another one, because these are your moving people, your moving vans. They're going to move your copies away from each other so that you can then constitute a nucleus around two separated copies of your DNA. So they're going to be moving. And a lot of times, by the time this happens, they've already taken up positions at opposite ends of the cell. Now, do cells have ends? Well, yeah, because once they get in position, we go, those are the ends, those are the poles of the cell. So they kind of define the ends, and since a globe has poles, what becomes the middle is then called the equator. Now we're into prophase. Prophase is the actual, let's get this all set up so that we can get the chromosomes away from one another stage. And a lot of stuff happens. Typically, early prophase looks different from later prophase. And sometimes when we do a mitosis lab, I'll have you try to find early ones and draw and label those, and then try to find late ones and draw and label those. A lot of times our slides are not good enough to really help you do that. So typically I just have you do prophase. But here's the stuff that's going on. We'll go back to the centrosomes. The centrosomes have migrated to different ends of the cell, the poles. And they start to spit out microtubules in every direction. So the screen, we'll call the screen the cell. And so here you've got centrosomes spitting out microtubules in all directions. Some of them go over and hit the membrane and stick there. These are your anchoring spindle fibers. These microtubules are called spindle fibers. And these guys are going to be pulling on things. They need anchors. So some of the microtubules will anchor against the membrane, and the other ones will radiate across the cell. So what's happening in the middle? These guys need to connect to your chromosomes. Right now, your chromosomes are not accessible because they've got a nuclear envelope around them. And they're not accessible for a couple of other reasons. So during prophase, the nuclear envelope goes away. It's sometimes broken into bits and pieces of membrane that are kind of spread away. It could be broken down into molecular pieces that will be reassembled later. You've got all the enzymes to do that. There's huge arguments for years over just what happens to the nuclear envelope. And like all of these arguments, it's both answers are right. It depends on the cell. Some cells completely disassemble, some cells completely fragment it, and a lot of cells do both. But your nuclear envelope is going away so that your spindle fibers can get access to your chromosomes. In the meantime, your chromosomes are getting skeined. If you know anything about yarn, that's what a blob of yarn is. That's a skein. Because if you're going to keep your yarn in a package, like in a box, you don't have it all unraveled, because then you'd never get anything separated from one another. That's true for chromosomes too. They need to be so tightly packaged that they don't tangle on one another when you're moving them around and trying to separate them out. So during prophase, they start to become visible under a light microscope. And that's was originally called chromatin, visible. They actually produce, they're slightly colored with a bright kind of light behind them, chroma for color. And later on, they discovered that these guys, that there was a whole complex of the chromosomes that was there all the time. And they went, well, we'll call that chromatin too. That won't be confusing, but it is. And so these show up under a light microscope. Sometimes it's very thick lines, sometimes it's very thin lines, but at least it's visible lines. And sometimes they get tight enough that you can actually see little XY kinds of chromosomes. A lot of times they just look like kind of ribbons, and seeing the connector points is kind of, sometimes you do, sometimes you don't. It all depends on the nature of the cell and the nature of the chromosomes. But chromatin appears, and once you start to see actual ends, even if you can't resolve the chromosomes completely, you're seeing chromosomes. Well, you're seeing chromatids, because they're still connected to one another. The copies are still connected to one another. So, okay, I'm a chromosome. Here's a chromatid, ignore my head. My legs are the other. And here's my very fat centromere holding on to the two copies that I've made. Well, the centromere that holds on is sticky for microtubule ends. So the nuclear envelope goes away. I get all packed down until I'm just kind of floating around there. And at some point, the microtubule end, a spindle fiber, catches me here. And that causes the microtubule to start being disassembled at the centrosome end, and it makes it shorter. So away I go toward that end of the cell. But in the meantime, I've turned and I'm gonna get hit by spindles from this direction. And they're gonna try to pull me that way. Well, if this is the cell, I'm going that way because the longer the microtubule, the harder it pulls. So this one's longer, I'm going that way. This one's now has to assemble more microtubule and get longer. So I'm gonna get to here, and now this one's gonna be longer, and I'm gonna get to here, and now they're gonna be the same length. They're gonna be pulling equally. They're gonna position me in the middle of the cell. That's happening during prophase. The microtubule is attached to the chromosomes once the chromosomes are accessible. The nucleolus goes away, because remember, chromosomes wind through the nucleolus and you need to not have them confined in any way. So the nucleolus is kind of unwound and disconnected from the chromosomes. And they wind up in the middle of the cell. So you're no longer in prophase. And you're now in metaphase, and I think we're going to pick this up on the next video.